Hello, bio people. Today we're going to be moving on to the topics of ventilation in both humans and goldfish. Before we dive in too deeply, I want to make sure we're all on the same page and that we all know that respiration and ventilation are not the same thing. Respiration is the physical gas exchange in and out of a cell, where oxygen ultimately becomes CO2 through cellular respiration. So almost all cells in your body are undergoing respiration right now. Ventilation is the movement of air in and out of an organism. So when you take a deep breath and pull air in and then exhale and expel the air, that's ventilation, not respiration. A good way to remember this is that a ventilator is a machine that they tend to use on patients who can't breathe on their own, and it physically pushes air in and out of their lungs. So if you hear a reference to somebody who is being kept alive by machines, or if they're thinking about pulling the plug on someone, this is usually what they're referring to. We're going to start our study of ventilation by taking a look at a set of human lungs. Here you can see the patient has both a left and right lung, and that directly underneath them is the diaphragm, which helps to push air in and out of the person's lungs. The first structure we're going to take a look at is going to be the trachea, which is this structure right here, and which you also might heard referred to as the windpipe. The trachea is reinforced with a series of cartilage rings, which help to keep it from collapsing if you take a really deep breath. You can feel your own trachea pretty easily if you look straight up at the ceiling and then rub the area on the bottom of your neck. If you do this correctly, you should be able to feel the cartilage rings in your own trachea. The trachea then separates into two distinct passageways, which are both called bronchi. You have a left and right bronchi that each lead to their own lung. When you get bronchitis, which is what you get if you have a really terrible cough and your chest hurts, this is generally because these two passageways tend to get very irritated and inflamed. The bronchi then split off into what we call bronchioles. The bronchioles are smaller than the bronchi. Finally, at the end of each bronchiole, you have a series of small air sacs called alveoli. I've only drawn 3 of them here, but you actually have many alveoli in your lungs. All along these passageways are a series of blood vessels. They start off as being pretty big, and they actually get a lot smaller as you move down towards the ends of the alveoli. So over here in the bronchi, they might be arteries and veins, but down in the alveoli, they would actually be capillaries. The blood vessels are going to explain how the gases get out of the alveoli and into the rest of your body. Now we're going to look at an alveolus a little bit more closely. Okay, so here are 2 of my alveoli, and to help us understand what these look like, I'm going to do a cutaway of one, and I'm going to show what the outside of the other one looks like. Okay, so we can see that this is actually a network of small bubbles that are called alveolar sacs. The reason that so many alveolar sacs make up an alveolus is because this increases the surface area inside the tissue of your lungs. The alveoli are nourished by a series of blood vessels. These blue blood vessels would be the pulmonary arteries, coming from the heart, carrying deoxygenated blood. The red blood vessels would be the pulmonary veins, returning to the heart, now carrying oxygenated blood. Because the arteries would be so big, they form a network over all of the alveoli, like this. The blood vessels carrying oxygenated blood also form a network over the alveoli. So something like this. Now I've only drawn this over 4 or 5 alveolar sacs, but it would actually cover the entire alveolus. When the deoxygenated blood carried by the pulmonary artery reaches a little alveolar sac, it drops its carbon dioxide and picks up some new oxygen before returning to the heart through the pulmonary vein. Now let's look at the cutaway drawing of an alveolus. The walls around the outside of the alveolus are very thin. This means that the distance for diffusion is very short. Carbon dioxide from the oxygen-poor blood will then diffuse across the membrane and into the empty space in the middle of the alveolus. Then it will travel up through the bronchioles, up the bronchi, through the trachea, and out of the person's mouth and nose when they exhale. When the person inhales and draws in new oxygen, the oxygen can then diffuse across the membrane and into the blood to make it oxygen-rich blood. This new blood can then be carried back to the heart and pumped out to all areas of the body. So let's trace this process from start to finish. Air comes in through the person's mouth and nose and travels down their trachea, also known as their windpipe. It then enters into the bronchi, and then the bronchioles, and then into the alveoli. This new air brings with it lots of oxygen. The carbon dioxide in the oxygen-poor blood will diffuse out of the bloodstream and into the alveoli. Then there is space for the oxygen to diffuse out of the alveoli and into the blood to make it oxygen-rich. We've essentially just described what happens in this section of this particular loop, the pulmonary loop. Once the blood has been oxygenated, it can move back into the pulmonary vein and then go into the left atrium, the left ventricle, and the aorta in order to make sure that all parts of the body and the heart muscle are fully oxygenated. We're also going to look at what happens to your alveoli under two different conditions. The picture at the top of the screen shows a normal alveolus with lots of surface area. If a person smokes lots of cigarettes for many years, they may develop emphysema, which means that their alveoli have a lot less surface area, and it makes it very difficult for them to get enough oxygen out of the air. This is why you might see long-term smokers walking around with a portable oxygen tank. This increases the density of the oxygen in the air they breathe, and thus makes it easier for them to get oxygen into their bloodstream. In a condition called cystic fibrosis, the alveoli fill with a thick glutinous mucus as opposed to the normal clear fluid that keeps your lungs clean. This mucus is especially susceptible to infection. In the past, a diagnosis of cystic fibrosis, an incurable genetic condition, usually meant the patient would die. However, with modern medical advantages, we can make sure that these people can live longer, much more comfortable lives. Finally, a fairly common condition is called pneumonia. This is where the alveoli of the lungs fill with fluid, which is why it can be so difficult to lie down to sleep if you have it, because it makes you cough. Alright, so now we're going to move on to goldfish ventilation, and we're going to see how that's different from human ventilation. In fish, water flows through the mouth, into the buccal cavity, and then over the gills and out underneath a flap called the operculum. This flap right here is the operculum. It's a protective covering that covers the gills to prevent damage. You can tell if a fish is ventilating by the fact that its operculum is flapping. We're now going to slice our fish open this way in order to look at what happens inside its body when it ventilates. Right now what we're looking at is a cross-section of the fish where the plane is parallel to the floor. On the left side of the screen you can see its mouth, and then on the top and bottom of your screen you can see its operculum. It has an operculum on both sides of its body, one on the left and one on the right, which protect the gill slits, which are right here. When the fish opens its mouth, water flows into the area called the buccal cavity. When the fish's mouth is open, the operculum slits are flush against the sides of the fish's body, meaning that the operculi are closed. The fish needs to push the water over its gills and then out of the operculum in order to extract the oxygen and remove the carbon dioxide from its tissues. To do this, the fish first closes its mouth. When it does this, the water is pushed out of the operculum openings, over the gills, and back into the water. As the water moves over the gills on its way out of the operculum, the oxygen is extracted and moved into the fish's bloodstream, and carbon dioxide is removed. We'll take a closer look at this in 1 second. We're now going to zoom in on these structures here on either side of the fish, which are known collectively as the gills. In this picture, we can clearly see the structures we call gills, and we can also see the fish's operculum opened right here. Notice that there appear to be many layers of gills on this side of the fish. This supportive structure right here is called a gill arch, and attached to it are many fibers that collectively extract the gases from the water. The red structures attached to the gill arch are called gill filaments. If we look even more closely at each gill filament, we'll notice that there are tiny hairs on them called lamellae. The nature of the lamellae makes the gill filaments a little bit fluffy and greatly increases their surface area, just like the cluster of bubbles in the alveoli of the lungs increase the lung surface area. The more surface area you have, the easier it is to do gas exchange. We're now going to look even more closely at the lamellae in order to see how gas exchange occurs in fish. In order to understand how lamellae work, we need to understand a concept called concurrent exchange. Here I've drawn two sections, one that represents the water and one that represents one of the tiny hairs on the gill filaments called the lamellae. Now notice that the membrane of the lamellae is very, very thin. This means that there is going to be a very short diffusion distance, just like there was in the lungs. The goal of the lamellae is to get oxygen to diffuse out of the water and into the fish. To do this, we have the water flowing in a different direction from the blood flow inside the gills. In order to keep the fish alive, diffusion of the oxygen must be occurring at all times. If it stops, the fish will suffocate. The blood that initially enters the gills from the back of the gills has relatively little oxygen in it. For the sake of argument, let's say it has about 60% oxygen. In order for the fish to survive, we need 100% oxygen in its blood, otherwise the fish's tissues will not get enough. The water that comes in through the fish's mouth is full of oxygen. It's 100% saturated. As it moves farther through the fish, it's going to lose oxygen, which will diffuse into the lamellae. The blood that comes into the lamellae from the fish is going to start off with very little oxygen, but will gradually pick up more and more as it moves past the end of the gill filament. When the blood begins to return to the rest of the fish, it has a very high oxygen concentration. The concentration of oxygen in the water runs in reverse. When the water initially enters the fish's mouth, it starts off with 100% saturation and then gradually decreases as the oxygen is sucked into the blood inside of the fish. When it finally exits the fish, it's going to have less oxygen than when it began. Remember that things like to flow down concentration gradients, from areas where there is more of it to areas where there is less. Because there is always more oxygen in the water compared to the bloodstream, the oxygen is always going to flow into the lamellae rather than flowing the other way. No matter whether you're in this area of the lamellae or whether you're in this area, the amount of oxygen is always greater in the water than it is inside the blood vessels. Because of this, diffusion occurs constantly rather than coming to a halt. In order to really understand this, we're actually going to flip them around and see what would happen if we had concurrent exchange, where both the water and the blood flow went in the same direction. Now let's look at what would happen if both the water and the blood were flowing in the same direction. Again, since oxygen likes to diffuse down the concentration gradient, it would flow really nicely on this part of the blood vessel right here, where the concentration gradient is very big. However, it would slow down towards the middle and end of the blood vessels because the concentration gradient gets a lot smaller. Eventually, it would come to a complete stop when the concentration of oxygen in the blood and in the water are the same. When diffusion is no longer taking place, it causes lack of oxygen to happen to all of the fish's tissues, and the fish will die. So, in order to make sure that diffusion is constantly happening, we use a concurrent exchange system, where the blood and the water flow in opposite directions. One more key thing. Surface area is key in biology. The more surface area you have, the easier it is to do gas exchange. This is why gills tend to be very fluffy.